Good evening, everybody. My name is Mark Schlissel. I'm a professor of immunology and the Dean of Biological Sciences. On behalf of the College of Letters and Science, I'd like to welcome you to our 5th annual On the Same Page program. The goal of this program is to welcome freshmen and transfer students to our intellectual community by inviting their participation in the consideration of an important topic or idea. To satisfy my curiosity, how many freshmen and transfer students are here in the audience tonight? Show of hands. Tell your classmates that you'll have to tell your classmates what they missed and tell them they can watch the lecture on the web and they'll be tested on this before they can graduate. Tonight's lecture, Looking for the Good News in Your Genome, is the first event of this year's On the Same Page program, supported by generous donors to the Letters and Science College Leadership Fund, as well as sharing in sponsorship with the California Institute for Quantitative Biology, QB3, and the Howard Hughes Medical Institute. I'd like to call your attention to a number of other upcoming events listed here on this slide. This marks the beginning of about a month or so of consideration of this topic of personalized medicine. Other events will include panel discussions on genes and behavior, direct-to-consumer DNA testing, fictionalizing the genome, the uses and abuses of genomic knowledge, and another keynote address by well-known bioethicist and attorney Alta Charo on the personal genetics and public angst. Each event deals with aspects of the broader topic of personalized medicine, the use of an individual's own genetic information or DNA sequence to better help prevent, diagnose, and treat disease. Like many new and evolving technologies, this presents a double-edged sword. There are issues of privacy, genetic discrimination, and many others that we need to consider as a society in order to take best advantage of this new set of technologies. It's a pleasure to introduce my friend and colleague from the Department of Molecular and Cell Biology, Professor Jasper Rine, our tour guide for tonight's trip through the human genome. Professor Rine received his PhD from the University of Oregon and did postdoctoral studies at Stanford before joining the Berkeley faculty here in 1982. Jasper is a world-renowned geneticist whose work has led to major discoveries regarding how the activities of genes are regulated by the way the DNA is packaged inside of cells, so-called epigenetic regulation, how metabolites of cholesterol are used to regulate the activity of proteins associated with cancer, and most recently, the impact of human genetic diversity on health, as we'll learn more about tonight. He's been a director of the Lawrence Berkeley Lab Human Genome Center and led the first mapping project that led to a map of the genome of man's best friend, the dog. He's also a co-founder of several biotechnology companies. Professor Rine is a former winner of the coveted Campus-wide Teaching Award, for which I'm personally jealous. And in recognition of his creativity and commitment to education, he was awarded a competitive Howard Hughes Medical Institute professorship, which he's using to remake the teaching of laboratory biology to our undergraduates. He's a generous servant of the Berkeley academic community, having served as head of our Center for Computational Biology and on the campus's budget committee. Amongst his many awards and honors was his election to membership of the National Academy of Sciences and the American Academy of Arts and Sciences. He's a sought-after speaker and consultant, helping transfer knowledge from academia into the biopharmaceutical industry. Professor Rine will speak, and then we'll reserve time for your questions thereafter. This lecture will be available on Berkeley's YouTube channel. It'll be available on iTunes University, as well as on webcast.berkeley.edu. Please join me in welcoming tonight's speaker, Professor Jasper Rine.
Phew, that's the part that always makes me nervous. Well, thanks to all of you for coming tonight. I have to say this is one of the great thrills for me of a career to be able to have this opportunity to lead off our discussion this year on personalized medicine. I've become really very much interested in this, and it's a real thrill for me to be able to be your tour guide for this evening. And because what I want to do is to give us a sense of trying to understand how variations in this iconic structure of DNA that you're all familiar with from CSI and various other venues have not only driven biological discovery since Mendel's peas, but also are now able to teach us things about our own genetic variation that we may want to know and that will enable us to do things that will understand ourselves better and I think to actually promote our health and to perhaps save the country from bankruptcy. So that's where we're going to go this evening. Now, before we start, let me emphasize the inspiration for this year's program came from the leadership of the College of Letters and Science. These are the deans of the College of Letters and Science who each year try to find a topic of contemporary interest whose significance impacts so many of the disciplines taught in the College of Letters and Science. Personalized medicine will touch each of our lives, from the 17% of the GDP that we're currently spending on healthcare to issues like who should control your access to your personal genetic information. Personalized medicine means nothing without personalized genetic information, and that's why I'm, as a geneticist, having the opportunity to present this story to you tonight. Now, our story can be usefully benchmarked as beginning in March 2001, with the completion of the first rough draft of the human genome sequence by a team of scientists from around the world at a cost of roughly $1.5 billion. Now, this discovery launched the new era of genetic research, but the hype had started even before the rough draft was completed, with Time magazine offering you for just $3.50 the chance to learn about drugs of the future, the amazing new medicines that will be based on your DNA, and the chance to find out how they will change your life. Now, Time, as the popular press often does, gets way ahead of the game, but what we'll see tonight is that, in fact, this vision is actually starting to be implemented. Now, you all know that one draft genome sequence is meaningless with respect to personalized medicine, and the goal of our discussion tonight will be to see how far we've come to enable you to understand what it means from all of your individual genetic variants. So let's take a look at your genome and think about what it actually is. So I love Leonardo's depiction here, his sketch, and I like to think of that as the hardware in which the genome is the software that makes it all work. So you're all made of 10 to the 14th different cells, many different types, and each of those types of cells has special properties that are guided by the gene sequences in those cells. On the right, we have a composite picture of the entire genome of a human being. And to remind you, each of you have pairs of chromosomes, 23 pairs of chromosomes, one that you get from mom, one that you get from dad, and you keep them in all of your cells through your entire life. Those of you with raptor vision may be able to recognize that this is the genome of a male. The way we know that is if we look at the last pair of chromosomes, there is the resplendent, glorious X chromosomes rich in genes and biology that females are endowed with 2 of, and guys, here's our runty little pathetic stub of the Y chromosome. That's why this guy is a male. So those of you that have problems with that, just get over it. It ain't going to change anytime soon. Now, one of the challenges in thinking about genomes is developing a sense of scale, because this is the stuff that we don't see and we have to infer indirectly. So let's try to develop that sense of scale. So a human cell has 7 picograms of DNA. Well, what does that mean? A penny is about 2.5 grams. That's not very much. So a microgram is a millionth of not very much, and a picogram is a millionth of a millionth, so you have 7 of those in each of your cells. Although that doesn't sound like much, if I were to take all of the DNA from one of your cells and line up those molecules end to end, it would stretch about that high. Actually, about that high. I seem to be shrinking recently. So what it teaches you is that this DNA must be incredibly tightly packed to fit inside the nucleus of each of our cells, which are about 10 millionths of a meter across. Now let's do a remarkable calculation. As I said, you have 10 to the 14th cells, more or less in each of you. So if I were to extract all of the DNA from you, don't worry, I'm not going to actually do this, but if I were, and line those molecules up end to end, the amazing thing is that it would stretch from the sun's surface out here distal to Uranus. So if you're looking in the mirror sometime and you're feeling kind of proud of yourself, indulge yourself, you deserve it. So what's the genome all about? Well, that's a piece of it. The human genome consists of about 3 billion letters written in the language of DNA, A, T, G, and C for the adenine, thymine, guanine, and cytosine. Now, 3 billion used to sound like a big number before the bank bailouts, but for biology, it's still a pretty big number. And what we want to see is what it would be like to try to analyze your genome sequence. So 3 billion consists of 1,000 letters like this written on 3 million sheets of paper. Okay? So if you wanted to read your genome sequence, the stack of that sheets of paper would be about 60 stories tall, and if you started reading it from one end to the other, it would be like reading the Los Angeles phone book 10,000 times in a row without stopping. Not a particularly useful use of your time. Now, these are genes. Medicine does not work on genes. Medicine works on proteins. Proteins come from genes, so let's think about what that relationship is all about. So here is the sequence of a particular gene. This is the gene that makes an enzyme called cystathionine beta synthase. How do we know what the sequence of amino acids in that protein is that's made from that gene? Well, it just turns out that a few billion years ago, something really important got worked out, which was the universal genetic code. There it is. It's the same in all organisms. And if we were to take a pencil and paper with that gene sequence in this table, we could deduce the sequence of amino acids in the protein, as I'll show you on this slide. Now, languages are written with different numbers of letters. You've seen how DNA is written in a language of 4 letters, Hawaiian is written in 12 letters, and proteins are written in a language of 20 letters, each letter corresponding to one of the 20 naturally occurring amino acids. So there's the glorious protein encoded by that gene. Doesn't look like much, does it? But left to its own devices, that protein can perform a computationally complex program of staggering complexity and fold into a 3-dimensional structure all of its own, the structure representing the positions of all of the atoms in that protein with a cute little molecule of vitamin B6 stuck right there in the center. So no computer can solve that problem at present, but every protein can figure out how to fold itself into that right shape. So there we've had it. We've kind of walked through the entire central dogma of molecular biology and molecular medicine, so if there's any questions on that, just save them for the end. So I told you in 2001, the first draft genome sequence was $1.5 billion. Let's take a look at what's happened in the next 6 years. These are the first 2 humans to have their own individual genome sequence completed. Here's Craig Venter, the head of the Celera project, and here's Jim Watson, one-time head of the US sequencing effort and one of the co-discoverers of the structure of DNA. There's Jim proudly holding his own genome sequence on his own little hard drive. His sequence cost about $1 million to obtain, and Venter's, we don't really know. He owned the company that did it, so he probably got a serious discount. Okay. Now, that was about a 1,000-fold improvement in efficiency over just 6 years, and the efficiencies just keep on getting better, and Berkeley actually plays a modest role in why. And the reason is this man, Dr. Stuart Blesson, a proud Berkeley graduate. He got his PhD here in geology, and Dr. Blesson, with his own funding, has established the Arkhon X Prize for genome sequencing, and he's offering $10 million to the first group to sequence 100 human genomes in 10 days for less than $10,000 per genome. And I think it's a pretty safe bet that this prize is going to get awarded this coming year. So the progress is continuing. There's the Personal Genome Project 10, led by this man here, George Church, out of Harvard University. And these 10 people are really extraordinarily brave because they've had their own genomes sequenced and have made all of it available to the public, to anyone with a computer connected to the internet. Why? Because they wanted to stimulate the development of tools for analyzing variation in genome sequence. And that's where the biology is all about. It's all about the variation. So that project was completed a little while ago. That's a $60,000 genome sequence. What's happening this year? Well, there's a project called the 1,000 Genomes Project. The goal is to sequence the complete genomes of 1,000 people of different races and ethnicities to give us a really molecular understanding of humanity. There are a couple institutions that are doing lion's share of this. This is the Beijing Genome Institute and the Broad Institute between Harvard and MIT. So what does it look like to have a machine that can sequence DNA? Now I'm going to show you one machine from one company, the Illumina HiSeq machine. Now I should say I'm going to speak about several companies tonight, and I should tell you that I have no financial relationship of any kind with any of these companies. Of course, I have friends at all of them, and one of the slides I will show you tonight is a reason why maybe you wouldn't want to have one, but I'm sorry about that, Francisco. I hope I don't insult you. Let's take a look at this particular machine. There it is. Kind of looks like a European dishwasher, right? But what it can do is extraordinary. It can sequence 104 human genomes per machine per year. The Beijing Genome Institute has 128, the Broad Institute has 50, And as of earlier this year, there was a backlog of 200 of them, 500 machines to be in the field next year generating 50,000 complete human genome sequences. Now, extrapolation is always risky, but if we extrapolate that the future will be less than the past, and imagine modest increases in efficiency, by the time the freshmen here are in their senior year, there will be 1 million people that will have their genome sequenced just by the machines, this one machine from this one company. Now, if that's not impressive enough, I can assure you there are numerous companies all ready to blow the doors off the capacity of that machine. Here's a good selection of them, and we are at ground zero for the revolution in next-generation genome sequencing. All of these companies are within a 50-mile radius of us. This is where the future of personal genetics is going to be written. Now, as I mentioned, an obvious question is, should I take my tuition for the spring semester and invest in one of these companies now? Well, before you do that, let me show you the next slide, which represents the plummeting cost of genome sequencing. You may be familiar with the famous Moore's law that shows how fast the cost of computing power is decreasing. Note that this is a logarithmic scale. Well, the cost of obtaining DNA sequence is plummeting at an ever faster rate. Now, if we use the remarkable analytical powers that are faultless from our colleagues on Wall Street, we can make a projection and show that by the end of next week, DNA sequencing will be free, and halfway through next year, these companies are going to be paying us for the opportunity to sequence their genome, unless, as they say on Wall Street, something changes. So what's the ultimate? The ultimate is the real personal genome project. I'm showing you here B. Reinhoff, here on the right, who is literally the poster child for personal genome analysis. Her father, Hugh Reinhoff, is trained as a physician, as a local entrepreneur, and he's trying to solve a very important problem. B was born with a connective tissue disease that has been undescribed before. It has baffled all of the experts in the world. And Hugh, being a very smart, very capable, very enterprising man, has taken it on himself to try to find the single misspelling in her genome that is responsible for her condition. And as of recently, he may have succeeded. He's also set up a website to help other people in that condition, that state, try to find the genetic foundation for the diseases in their family. So genetics has gotten to be the ultimate of personal. So that's all about variation. So let's take a look at variation. And let's imagine that this is my sequence, and look right here at that particular C. Can you see that C? All right, let's look at that sequence close up and compare it to what Dean Schlissel's might be. So there's mine, and there's the Dean's. Wouldn't you know it? I get a C, the Dean gets an A. Well, that's the way it is. So how much sequence variation is there? Well, it turns out we have a very good estimate for how rapidly this accumulates, and that number is 10 to the minus 8th mutations per base pair per generation. That's a little hard to appreciate until you multiply it by the number of people on the planet, and the product of that calculation teaches us something astonishing, which is that in every generation, all possible simple mutations occur at all positions in our genome. We're all mutants, right? And all possible mutants are made again every generation. So there is nothing we can learn about any sequence variation that is not relevant to somebody. It might be 2 people in Ulaanbaatar, it might be 50% of the planet. It's all significant. So we're going to explore this strange world of genetic variation. And if you're going to go to a strange place, it's awfully nice to have a guide. So if you were going to New York City, a Lonely Planet guide would be useful. There is a guide written to the human genome. It's a short guide. It's appropriate for sort of the senior undergraduate level, not quite yet ready for the freshman level. But more importantly, we need principles to guide our exploration of genetic variation, and there are only 4 of them. Here they are. The first one you, of course, know about, the principle of evolution by natural selection as the gift to us by Darwin and his contemporary Wallace. There is nothing in biology that makes sense except in the light of evolution, and I'll show you a really dramatic example that brings this home to variation. Here's the sequence of a gene known as the Werner's gene, abbreviated WRN, and I call it the ageless aging gene for reasons that you will see. So here's the sequence of a portion of that gene written in the language of proteins. The sequence of that portion of that gene has not changed in dog, mouse, or chicken, or fish. And it's only changed a little bit if we look in worm, fly, rice, and yeast. Yeast? Yeah, yeast, the stuff that makes the bread and wine that we're so fond of. Now here's something astonishing. If you change one of those letters there in a human being, this happens. That's a 6-year-old boy who's got the physiology of an 80-year-old man and who's not expected to live past 12. Now here's something really astonishing. If I make that same change here in the yeast gene, the yeast dies prematurely. So Darwin was more right than he could ever possibly have imagined. Not only is evolution conservation of function, it is conservation of function of profound importance to all of us, even down to the mechanism of aging in individual cells. So that's principle number one. Principle number two has to do with the evolution of humans and our migration out of Africa. Modern humans all evolved in Africa some time ago. Africa is the source of the richest genetic variation in humans on the planet. But about 50,000 years ago, when the population of Africa was about 10,000 people, small groups started migrating out of Africa to different places. And because we're sampling small groups out of a big pot of variation, where these peoples went, the resulting frequencies of those variants can be very different from place to place and very different from Africa. We'll use that principle later in our discussion. The third principle, you of course are all familiar with. It's the family structure, the pedigree. I'm showing you here a family of 2 grandparents, their 5 children and assorted siblings, and their 9 grandchildren. And you'll recall that, of course, that this fellow right here gets 1 chromosome from dad, 1 from mom, as do all the others, but of course the genetic deck is shuffled each generation. And so they get 1 chromosome, but the genes that are on that chromosome are kind of mixed up between generations. So this fellow has half the genes that his father had. This one has half the genes, but it's more or less a different half, and so on. This one has a quarter of the genes that its grandfather on that side had. And so you can see that if we were to analyze the shared genes between this quarter of the genome and that quarter of the genome and that quarter of the genome with a relatively modestly sized family, we could zero in on the genetic cause of the color red, okay? So that's the principle that's used to link genes to traits, and linkage is a powerful, powerful statement because linkage teaches us causality. We can figure out what gene variants cause traits. Now, that sets up the fourth and last principle. And this last principle is genetic guilt by association. It's very common these days, especially in the form of what's known as a genome-wide association study. It can be very powerful, but is the most frequently misrepresented genetic analysis in the press. So let's look at the strength of this, and then we'll look at the weakness of this. Its strength is utterly intuitive. Suppose you have a rare condition for which there is not a large family in which it's segregating. Well, you simply get a collection of healthy people, and then a collection of those with a disease that you're interested. You try to make those collections as similar as possible otherwise, and you simply scan across the genome, sampling sparsely at each position to find out what genetic differences distinguish one group from the other. So you might find that at this position on the same chromosome, all these people have, except one, have the A variant, whereas those with the disease has the G variant. Now, if the numbers are large enough to be statistically significant, as an aside to the freshmen, you must take a statistics class while you're here if you're to have a full and righteous life. If the data were statistically significant, you would be able to make the claim that this disease is associated with the G form of that gene. But is it causal? Maybe, but more often not. Why? Because we have only sampled sparsely across the genome. Genes travel in packs. It could be the gene over here that actually has the variant that's responsible for that. So association can get us in the vicinity. Association rarely gets us causality, although there are some examples of that. These studies are hard, they lead to misinterpretation, and never worry about an association study's result unless it gets replicated. So I'll prove that to you because we're going to map a very rare genetic disease tonight, right here in this room. Here's the disease. It is haggis eating. This is my hypothesis. Are there any haggis eaters in the crowd? 1. You can see it's a very rare trait. So what is haggis? Well, it's a little odd. It's a Scottish dish that you make by chopping up the intestines of sheep or calves. You mix it with lard and oatmeal and seasoning and boil it up in the animal's stomach. Anybody want to dash out and grab some? So we've already determined this is a rare trait. So our hypothesis is that there is a disease condition that causes the eating of haggis. So how are we going to find this disease gene? Well, it's simple. We go someplace with a suitably large population. Beijing will do. And we set up a booth that says free gene testing for all haggis eaters. Now, can you see these individuals identified by these red letters, arrows? No, but if you could, you would notice that they've got a reddish hair and they're speaking to the body we lost beside them. So we get a group of people that come up and say, hey, I love haggis, test my genes. So you get a little saliva sample from them, and you take a good number of matched controls that are non-haggis eaters. You'll look at the variants across the genome, and what do you see? Sure enough, you find strong genetic associations that are common to the haggis eaters and the non-haggis eaters don't have. So you conclude that I have found the gene for the haggis-eating disease. You also notice something odd about this disease. It causes a very strange fashion sense and tends to cause light skin, red hair, and a fondness for unusual, out-of-tune music, right? So is there a haggis-eating gene? Of course not. What we've simply found is a genetic difference in the allele frequency between Chinese and Scots. So this is a ridiculously simple example, but it illustrates a very important principle about how hard it is to know whether there's underlying structure and complexity in your population. And this is why association studies are very interesting, but have to be taken very cautiously. So we've learned a lot from the analysis of individual genome sequences, and so let me summarize what that is. Of course, there's 3,000 bases in our information, and in fact, there are 3 million genetic differences between any two of us. So we are 99% identical in all pairwise combinations of people on the planet. Now, those differences are interesting because the genome encodes 22,550 different proteins, which are the potential targets of medicine. Maybe 1,000 are actually used by the existing medicines. And there are expected to be 2,000 of those 3 million differences that affect the structure of those proteins. And our best estimate is that 120 of them are expected to be deleterious. Now hold on to your seats now. And 5 of them are potentially lethal in all of us. So it's a good thing our genes come in pairs and we've got a spare. And interestingly, we get about 80 new variants each generation. So the 3 million differences between you and everybody else, a few of those you rolled yourself, but most of those you inherited from your ancestor. So what do we do with this kind of information? Well, it creates a central dilemma that was best described by Mark Twain in the context of something else, which is that everyone complains about the weather, but nobody really does anything about it. So here's the issue. We've all got problems in our genomes. Is there something constructive we can do about knowing what those issues are? And here's the problem with personal genetic information. There is the perception that it is bad news. And I want to argue that mostly that perception is wrong. I mean, that's going to be a take-home message from our discussion tonight. But one part of it that is right is that learning your genetic information can invade someone else's privacy, and genetic privacy is something that I think is really, really important. Now, how does that work? Well, let's consider the following pedigree. Here's a pedigree with a grandmother that has some late onset condition. Alzheimer's would be an example of that. The mother is taking care of this grandmother and certainly is worried about her own risk for Alzheimer's, but simply does not want to know. Now, in only a small number of cases is Alzheimer's simply inherited, but there are such cases, and there are gene tests for such cases. So if this is any of you, enterprising young Berkeley students who know about all this stuff, you might say, gee, I want to know about that variant so that I can do something smart like buy long-term health insurance while it's cheap and still legal for me to do this with taking advantage of genetic information. But if you find out that you carry that variant, your mother must carry that variant, and unless you can keep that message secret, tough, your mother's going to find out something she doesn't want to know. You will have invaded her privacy. And frankly, there's not a very good solution for that. So that's just part of the lay of the land. So let's now turn our discussion in a more positive way and think about what would good news in the human genome look like if we could find it? Well, it ought to have several qualities. It ought to be non-frightening, for sure, actionable, safe, inexpensive, and effective, things you could do about it. It should have minimum privacy issues, and it should improve your well-being. And I think that nutrition and its intersection with personal genetics will be the low-hanging fruit, the gold mine of where we're going to find most great good news in our genome. This idea does not come from me. This idea comes from my great friend and colleague, Bruce Ames, who's shown here, receiving one of many honors. Bruce is responsible for many, many, many important discoveries, but his perhaps most obscure paper, published when I was a freshman, makes this startling claim. 20% of certain inherited diseases of bacteria are vitamin B6 remedial. If that sounds obscure to you, well, it is. It's taken me about 40 years to realize how important it is. I think this paper is fundamentally foundational because it teaches us that there is genetic variation that can be ameliorated by something simple that we can do with our diet. Now, is it possible this thing applies to humans, this principle? Well, what do you think these animals have in common? All primates, guinea pigs, and the lovely fruit bat. Any ideas? Well, you've never had to feed your dog or cat vitamin C because your dog or cat can take glucose, a form of sugar, and with biochemical transformations, make vitamin C. Your dog or cat is not at risk of scurvy on long ocean voyages. It doesn't have to eat citrus. But each of us is at risk for scurvy, as well as guinea pigs and fruit bats, because we all have defects in the gene for the enzyme that catalyzed this last step. So we are all mutants that require exogenous vitamin C. So could this apply also to other variants? Well, this is an extreme case. Our gene is just dead, ain't never coming back. But here's the human metabolic wiring diagram. So metabolism describes the chemical transformation reactions of biology. There's about 10,000 of them. This is a very highly stylized schematic. Each dot represents a chemical that's connected by a line, which is an enzyme. There's about 10,000 different reactions, each catalyzed by an enzyme, and 640 of those enzymes need a vitamin or a mineral as a cofactor. So Bruce has written persuasively about the importance of genetic variation in each of these enzymes for our optimum well-being. I've been so impressed by that argument that, in fact, my lab has turned its attention to trying to find out which of these variants are actually having that kind of impact, and frankly, we're finding a lot of them and having a lot of fun doing it. But what about you? How could you go about learning about your personal gene variants? Well, it might surprise you to know that it's been happening already. If you were born in California, just a couple days after you were born, your heel was jabbed by some physician, ouch, right? And your blood was extracted, dried onto these kinds of cards, and analyzed by methods not as reliable as gene testing for 30 different inherited disorders with very significant medical consequences. Why? Because if you were born with any of these disorders, there's something that can be done about it through changing your diet and other things. So if you're worried about genetic testing, forget it. The horse is out of the barn on that one already for anyone born in California. So let me pose a provocative question. Should college students be subjected to mandatory non-anonymous genetic testing for serious medical conditions? If I were to take a poll, my sense would be the feeling here is probably, gee, not a very good idea, is it? Well, it's already happened. This year, in April, the NC2A announced that requirement for all Division I college athletes to be tested for carrier status for sickle cell anemia. Why? 8 college athletes over the last few years have died during heavy workouts with the sports teams because, unbeknownst to them, they were carriers for sickle cell anemia. So it is now required for a Division I school to have all its athletes tested unless the athletes sign a waiver promising that their estate will not sue the university should they die at football practice. Okay. Now this brings us to the issue that's become highly contentious of direct-to-consumer genetic testing. This is an unfortunate situation right now in which the Food and Drug Administration, the American Medical Association, and legislatures are sparring off against entrepreneurs, consumers, and the right of each of us to know about our own genetic makeup. And this is an unfortunate situation, but it is not uncommon when technology and knowledge outpace understanding. Now, to be sure, there are companies out there, it's not hard to find their websites, that are the modern equivalent of snake oil, that offer genetic tests for things that are completely outside the range of science. But Francis Collins, the director of the National Institute of Health, sent his own DNA sample to 3 companies, leaders in this technology, and asked them to analyze his sample anonymously, and he compared the results to see how good the results were for these 3 companies. And his view of this, which he's published, is that the overlap in the data was extremely high. So the quality of the primary data from the leaders in this technology is very, very good. He has quibbles over the interpretation, and we'll come to some of those, but the leaders in this technology are doing a scientifically good job. So to get a better picture of this, I decided to have my own DNA analyzed in preparation for this lecture so that I could give you, so to speak, a peek inside my genes. So are you ready for this? Within limits, right? Okay, here's Jasper's disease risk profile. Whoops, some of this is off the screen, but I think we can get it. So here's a list of diseases, and here are some numbers that are solid numbers. This is the average risk for people in the population of these diseases. And these are other issues which are not as solid as these, so confidence. The confidence refers to how sure are the companies of their call, and confidence is sort of an expert opinion based upon hard statistics, expert reading of the literature, and good, solid judgment. So that's subjective. The risk profile is somewhat subjective, but to a first approximation, it's useful. And so let's look what's in store for me. Now, I was quite surprised to find that I think most of this is good news that I can actually do something about. All right, psoriasis. I got a 4-fold elevated risk for psoriasis. Not a lot I can do about that, but that probably explains why my hands tend to peel from time to time, and it's nice to know. Atrial fibrillation. Well, it's a modest increase in risk, but this has caused me to drag my colleague Mike Botchin out to the gym with me every once in a while just to make sure that my heart is ready. Age-related macular degeneration. This is a common form of blindness in the elderly, and even though I am not even close to being elderly, when I got this information, I immediately made a long overdue appointment at the optometry clinic here. And I am sure that when I tell Dr. Peng about this, he's going to look a little closer at my macula to see is there something I need to be concerned with there, and I will watch the literature on research on macular degeneration. And melanoma, very serious disease. Now, my risk is only modestly elevated from the population-wide risk, but I went, geez, that's awfully high population-wide, and this has really got me to make sure I use my sunscreen when I go sailing. So that's what my risk profile looks like for overt things that I'm going to face, but what about my carrier status? What variants am I carrying that might affect any of my offspring? Now, there's a guy in the second row that's looking awfully nervous right now. That's my son. He hasn't seen this data yet, so Jeremy, hold on to your seat. I don't want to know. Close your eyes. Whammo! I've got one. Alpha-1 antitrypsin deficiency. This, when present in 2 copies, is a very serious disease, especially for people who are smokers or who work in dusty environments like mining and milling. I've got 1 copy of that, no particular problem. But interestingly, my grandfather died at about my age of severe lung complications. He was a lifelong cigar smoker, God forbid, and he also worked as a cabinet maker in a dusty industry. That may well be where my carrier status came from. So, Jeremy, it was a really good idea you didn't take up smoking. Now, what about the rest of these? Well, you might look at this variant absent and say, hey, man, that guy's lucky. He dodged the bullet. But did I? So let's focus in on one pair of these genes, BRCA1 and BRCA2 that have been in the news. These are genes in which carriers are at much elevated risk for serious diseases like ovarian cancer and breast cancer. Now, to be sure, my risk of ovarian cancer is 0. And as a male, my risk of breast cancer, even as a carrier for such traits, is pretty low. So I know that, but why did this company say that my variants were absent? Well, this company is very good. If you actually click on this information, what you discover in the fine print, or not so fine print, is that only 5 of the hundreds of possible variants for breast cancer have actually been tested in my DNA, and I have none of those, but God knows if I have any of the others. All right? That's sort of an interesting point, and that also brings home the fact that I may well be carrier for any of these other traits, but I simply don't have the most common allele that's looked at. Because this company, and this is one of the best companies, looks at only about 500,000 possible positions in my 3 billion letter genetic code, so until we have our own individual complete genome sequences, we'll never really be sure. The last class of personal information that we'll talk about tonight has to do with pharmacogenomics, and this is a situation with respect to drugs in which one size decidedly does not fit all. Now, it is a sad but not very widely appreciated truth that if you have a collection of patients with exactly the same diagnosis and you give them exactly the same prescription, only a subset of those will benefit from that drug without toxic side effects. There'll be another group for which the drug is not toxic, but it doesn't do any good. There'll be a group for which the drug is toxic, but at least somewhat beneficial. And there'll be the very unfortunate ones which get the toxic effect, but not the beneficial effect. This sounds crazy. That's what the FDA is supposed to protect us on, isn't it? But it's true. I'll show you a dramatic example of why with one drug called Coumadin. Now, there is an excellent chance that relatives of many people in this audience have been prescribed Coumadin. Coumadin was prescribed 34 million times this last year, and it is used as a control for blood clotting in the case of strokes, heart disease operations, and various other conditions. So you have learned in high school that blood clotting uses vitamin K. Vitamin K is modified, and in its modified way, turns proteins that are in our plasma that are not clots into clots, and is used up in the process and needs to be recycled. But Coumadin inhibits the recycling of vitamin K. So what's the big problem? Well, the problem is that Coumadin is also acted upon by numerous drug metabolizing enzymes in which there are common and serious mutations that affect their effectiveness. As a result, the effective dose of Coumadin that you actually receive, if you consume the same amount of it as somebody else, can vary by 20-fold. And Coumadin is one of the 10 leading causes for the 1 million adverse drug reactions in clinical settings in the US every year, okay? So let's look at my genes, what's in store for me. Well, lo and behold, I am in fact of heightened sensitivity for Coumadin. I'm not taking Coumadin, but you can bet this is information I'm going to give my doctor should I ever have to take a Coumadin prescription, because what would be normal for somebody else could cause serious bleeding disorders in me. So that's a peek inside my genes, and now let's turn our attention to our program here this year, the On the Same Page program. Well, by now you all certainly know the nature of this program. We decided to do an experiment, and rather than having you read a book, to give you the opportunity to participate for the first time at any campus, at anything like this scale, the opportunity to have 3 common gene variants tested in you in a completely anonymous, completely voluntary way, okay? And 726 of you chose to do that with properly filled out consent forms. Now, why did we decide to do that? Well, what we wanted to do was to stimulate discussion, as we knew this would, among the many different voices and concerns and levels of expertise in the College of Letters and Science, because we think this is really a fundamentally systematic issue that our society needs educated people to deal with. We also felt that it was really important that students have a personal connection with a complicated subject, to have the desire to really learn deeply about it. And we don't know if that idea is right. So all of you will be getting a survey asking you whether or not this experience increased your interest, increased your connection to the material, did it help you learn more about it or not. Please fill out that survey. It'll help us know to what extent this experiment was successful and should be or not be replicated someplace else. Now, as you may have heard, this program got a lot of attention. Note the date of the first article in the New York Times, May 18th, just a few days after the program was announced. This is a pretty good article describing pretty accurately what we were intending to do in mostly flattering terms. Now, we've gotten a lot of press, and some of it has been really gratifying, including press by Professor Russ Altman, who I've discovered is the smartest professor at Stanford. Now, there's a lot of smart people at Stanford, so how do I know that Russ is the smartest one? Well, it's easy. Look at what he wrote. Yeah, how about that? This may be the first Stanford professor smart enough that we should try to recruit him to Berkeley, but let's take a look at what he said. So he describes our program. He says, I believe it is absolutely critical that we immediately start educating students about genetics and the use of genetic tests in making decisions in life. I think that direct engagement with personal data is one very effective way to make all the issues crystal clear. And now, imagine the pain this poor fellow wrote as he typed these last lines. So at least in the narrow arena of providing genetic information so students can learn, go Cal. Thank you, Professor Altman. So what do we want to do with your samples? Well, we decided that what we wanted to choose, let me back up for a second. The choice of what genes to test would be different no matter who made that decision. Our thinking was the following. We wanted to test something you probably already knew about yourself. We wanted to test something that you were likely to learn about yourself in the future. And we wanted to test something that we thought was probably pretty safe, but which there is conflicting information in the literature to give you an experience of dealing with conflicting information. So here's what we did. One of the genes was lactase. Now, lactase is a fantastically interesting gene with respect to human anthropology. It's the gene that makes the enzyme for digesting lactose, the sugar in milk. And lactose intolerance in adults is the normal condition of humanity. Most people as adults cannot metabolize lactose. That's why you feel kind of uncomfortable and bloated when you have a milkshake, okay? But it's descendants of Northern European herders who, for obvious reasons, had the advantage happen to them when they obtained a mutation that let lactase stay on as an adult. How did that work? Well, this is a cartoon form of the gene, and this is the part of the gene that encodes the enzyme lactase, and written in DNA near that gene is a genetic switch that can be in the on or off state. And these descendants of Northern Europe have a mutation which breaks that switch and traps the gene in the on state. So that's lactase persistence. Variation in ethanol metabolism we explored by studying this enzyme, aldehyde dehydrogenase. When you turn 21 and have your first alcoholic beverage, your ethanol will be converted in 2 steps to acetaldehyde and then to acetate, where it will enter metabolism at large. Variation in this gene has nothing to do with your ethanol tolerance per se. That's this gene. But aldehyde dehydrogenase controls how quickly you metabolize acetaldehyde, and if you have 2 copies of the variant that makes that metabolism slow, when you have your first alcoholic beverage, you will find yourself flushing, your face will turn red, the blood will rush to the surface, and it will be an unpleasant experience. One version of that gene, you will flush less well. Interestingly, people who have 2 copies of that variant have a very low risk of alcoholism because the experience of drinking ethanol is so unpleasant. Now, as an aside, there are a few associations that suggest that one of these variants is associated with elevated risk of a rare cancer, esophageal cancer, in people who have that variant and who drink heavily and smoke heavily. The risk of that rare form of cancer goes up about 3-fold. That elevated risk is still a small fraction of the total risks that come with heavy smoking and heavy drinking, but that's something to know. The third variant that we tested has to do with the metabolism of folic acid. So folic acid comes from leafy greens, which your mother told you should eat. It enters metabolism when we consume it, and eventually encounters this enzyme, methylene tetrahydrofolate reductase. Methylene tetrahydrofolate reductase uses folic acid to convert homocysteine, which is an odd, non-natural amino acid, to something natural. So what does all this mean? Well, there's a story about the methylene tetrahydrofolate reductase gene that has been written in more than 1,000 research papers focusing on a very common variant in which the cytosine at position 677 is converted to a thymine. Some studies have shown a weak association with birth defects like spina bifida and coronary disease. But larger, more recent studies have been unable to replicate these. So this is a classic dilemma. What do you do when the scientific evidence is mixed? Well, you have to understand what the nature of a scientific publication requires. Your data in support of your hypothesis has to be good enough that the chance of getting those data by chance is 5% or less. That's the standard. That's the bar you have to clear. So at that level of statistical significance, see why you've got to take that class? At that level of statistical significance, we expect 1 in 20 conclusions to be wrong. 1,000 studies, some are going to be wrong. So what do? Well, on average, you're better off trusting the newer data from larger studies as a way of easing your mind. The other thing you can do to ease your mind is good, simple quantitative thinking. So let's take a look at this allele quantitatively. Let's call its frequency in the human population, which is 0.3. We'll call that value Q, and that allows us to calculate rather easily the frequency of not being Q, which is 1 minus Q, or 0.7. Have I lost anybody yet? Good. We'll call this P. Now, if you reach into the dark recesses of your memory from high school biology, you'll recall the Hardy-Weinberg equilibrium, and you wondered, what was that thing ever useful for anyway? Well, here's what you use it for. You can calculate carrier frequencies, and you can calculate the frequency of people that have only 2 copies of a particular gene variant. Here's the calculation. 2PQ, 2 times 0.7 times 0.3, 42% of the people on the planet are walking around with 1 copy of that variant, and the number that are walking around with 2 copies of that variant are 9%, okay? So more than half the people on the planet carry 1 or 2 copies of that variant. If that variant were responsible for significant disease on its own, we would know that by now. So should you worry about it? Interesting question. You could say, well, maybe this is the prototype of other variation that has not yet been discovered in other genes affecting metabolism, and maybe it's a really good idea to eat a good balanced diet. That's what I do. I also actually take a daily vitamin supplement. What the heck? Who knows what else is in my genome? So this brings us to the process by which our project was launched. Now, because the Bring Your Genes to Cal program was, the gene testing part of it was strictly educational, we did not really have to seek approval by the Committee for the Protection of Human Subjects. But we thought because this was bound to be controversial, and because we wanted to have best possible practices and benefit from the experience of the experts on campus with the responsibility to approve or disapprove any procedures involving human subjects, we sought that approval. We submitted an application on March 11th. It was a detailed 37 pages. It was reviewed by these experts. They made very useful suggestions. We revised and made amendments, and that led to the consent form that was sent to all of the entering students in the College of Letters and Science, giving you the opportunity to decide to participate or to decide not to. We think both decisions, as long as you made one of them, was a success for our program because we wanted people to think about this deeply. We also obtained funding, and I'll explain that more, from non-state funds to pay for this so that we couldn't be accused of spending our strained UC budget on what some people didn't think was a good idea. So let's take a look behind the scenes at what we actually did. While we received 726 properly consented saliva samples, we, in this case, I have to say, is the Lisa Barcelos Lab and the School of Public Health, who are the heroes of our program tonight. So they took those samples, the consent forms were separated from them and were shredded immediately, and then every other thing was directed to the barcode which those of you sent in samples put on that sample. So here the barcodes are being read. The Barcelos Lab purified DNA from all that spit, right? So they measured the DNA concentration, and using cool liquid handling robots, they adjusted the concentration for the tests of gene variants. I'll show you on the next slide the way that test works. It's slightly technical for those of you like me, my geeks, my fellow geeks. For those of you that get lost, don't worry, you won't need to know that. Here's how it works. Consider a position at where we're trying to decide whether you have the G or the T variant. So this is your DNA in purple, and you have the G variant. We don't know that. We want to know if it's G or T. So what we do is to mix your DNA with a synthetic piece of DNA that has a C right there and a red tag. We also mix it with a synthetic piece of DNA that has an A right there with a green tag. Now that A does not pair with G, you remember that, but the C does. So if we then add an enzyme called DNA polymerase and subunits of DNA that are labeled with a fluorescent marker, we make new DNA here, but not there. And then we mix these tagged DNAs with special beads that stick either the red tagged or the green tagged product of that reaction, and then we scan it in little microtiter plates. Here's a picture in the lab of setting up these hybridization reactions, and here's a picture of one of the commercial scanners that we use for doing this kind of analysis. And then to protect your privacy, we removed the barcodes from the tubes once the DNA was analyzed. We destroyed all of the DNA chemically by soaking it in bleach, and then we wrapped it all up in biohazard bags and sent it to an incinerator in San Leandro. Nobody has any chance of getting to any of your DNA. Now, I don't have time to do the next slide complete justice, but in any procedure, any laboratory test, there is always the opportunity for error. And the Barcelos Lab practices this craft at the highest level of perfection. My physician tells me that 10% of the lab tests she sends out is obviously an error. Let's take a look at what the Barcelos Lab can do. So on each plate, there are negative controls where no DNA was placed. There are family controls to see if we could make sure that we could always tell a positive when it should have been positive and a negative when it should have been negative. There were other positive controls. There were within plate replicate controls. There were between plate replicate controls. And we even took a little bit of DNA from 250 of the samples and sent it to a lab at UCSF, which uses a slightly different technology to make the calls. And the data are stunning. There was 100% agreement in every place there was a control with what we expected. That does not say that there's not an error someplace in here, but it puts that error rate in the vanishingly low, low rate. Much better than what my physician gets. Now, interestingly, there were 2 sample failures. 2 samples from which we did not get any genetic information. Now, considering the creativity of the entering class, I would be seriously disturbed if at least 2 of you didn't manage to get your family dog to drool in that tube just to see if we could tell whether you're gooning us or not. So I think that's probably what happened. So what did we learn? Well, let's take a look at the entering class of the College of Letters and Science this year through the window of the DNA samples from the 726 participants. Here is the MTHFR gene involving folic acid. Just like we expect around the world, 9% of people, more or less, have that same variant, which makes the enzyme slightly less functional, and about 40+% carry that variant. I don't think any of these people should worry about anything, frankly. Lactase persistence. It looks like 20% of you will be able to enjoy milkshakes at Oscars till the cows come home with no discomfort. Obesity may be a different issue. Probably 40% of you will have some discomfort, and the rest of you, well, you've seen your last good milkshake. And ethanol metabolism. The results here were particularly interesting to me. The people with 2 variants, the serious flushers, are only a small percent of the people who have participated in the study. Modest flushers are about 15%, and 90% of you, when you have your first ethanolic beverage, will not flush, okay? Now, our plan initially was to provide the individual data, which would be much more satisfying to all of the participants in this, but we hit a roadblock along the way. So what was the roadblock? Well, remember that date that I told you about the first article in the New York Times, which I think was May 18th? Well, not very long later, May 19th, the picture had changed. The tests were being criticized. Now, these criticisms came in several flavors. We'll just talk about a few of them. One class of criticisms, and it's almost not even fair to call them criticisms, were issues that were raised by our faculty colleagues in different disciplines. Many of these ideas we thought were terrific ideas, and we incorporated them, and the project evolved during its execution. So I want to thank those people for those kinds of criticisms. There was another class of criticism that largely came from the misunderstanding of the project, as it was fairly inaccurately portrayed in some of the hundreds of articles written about this. There was another class of criticism that I think fundamentally represent what all of us can understand, the sort of discomfort with new technology that moves too fast. I mean, how many of you enjoy upgrading your operating system, right? And then there was the other class of criticism that probably we won't get to. Okay. So this criticism, though, was really terrific because it did exactly what we were wishing for. In fact, it fulfilled our wildest hopes that we would get people from all disciplines talking about this, debating it, and sort of engaging in the kind of intellectual exercise that the College of Letters and Science is designed to promote. But we even got criticism and debate that sort of was more than we wanted, and here's an example of it that comes from the perception gap between what we were doing and what people thought we were doing. Here's a no doubt well-intentioned assemblyman who introduced in a bill, Assembly Bill 70, into the California Assembly, whose goal was to make this project illegal and to fine the university with its general budget from all of the money that we spent on it, even though not a nickel of taxpayers' dollars was spent on it. I didn't actually expect that. So I had a chance to spend 3 days in Sacramento this summer. And the hero of this experience is this fellow, Steve Juarez, the Associate Vice President in charge of State Government Relations. Steve is among the most impressive people I have ever met. Steve understands the legislative process completely. He knows many of the legislatures, and he took us in hand, a delegation of us. We met with the author of the bill, we met with the Assembly, we met with the Senate, and by the time the smoke had cleared, the bill was dead. That was good news. We had 40 minutes of ecstasy, until we encountered the California Department of Public Health. Now, the Department of Public Health is charged with optimizing the health and well-being of the people in California. That's great. And they have the authority to enforce laws involving clinical testing. And among the laws are the Public Health Service Act, importantly, as amended in 1988 by something known as CLIA, the Clinical Laboratory Improvement Amendments. Now, these are a good thing. These amendments require that any company that provides a clinical test on any sample has to have proved competence, has to maintain good records, and can only do tests that are ordered by your physician, and the results are only reported to your physician. They don't deal with consumers, okay? So you might think, well, a genetic test is sort of a clinical test, so why did we ever imagine that we could do this in the first place? And the answer is really very straightforward. Clearly written in California law is the following exemption. And I'll read it to you. The law exempts from regulation those laboratories or personnel, and here's the critical parts, that perform clinical laboratory tests or examinations for research and teaching purposes only. That's what the On the Same Page program is all about. And do not report or use patient-specific results for the diagnosis, prevention, or treatment of disease. Well, we don't actually think the entering class are patients of the University of California, but if they are, we're certainly not diagnosing disease. Or for the assessment of the health of an individual. We're not assessing your health. We're assessing the variant that you have at 3 positions out of the 3 billion in your genome. Now, if you find that argument that I just made persuasive, you're in serious trouble because you should never trust a genetics professor to interpret law for you. But we had experts on our legal team, including Rachel Nozowski, the principal counsel from the University of California Office of the President, Christopher Patty, UC Berkeley's chief counsel, and Pat Schlesinger, also a lawyer, who's the assistant vice chancellor for research administration. Amazingly to me, they read the law the same way that we did. So with their help, encouragement, and guidance, we sought a meeting with the California Department of Public Health's leadership. We explained our position, confident that they would grant us the exemption that we needed to return the data to you. And Dr. Mark Horton, the director, said no. Now, I was rather baffled by that decision, as baffled as this other gentleman seems to have been on another occasion. We have asked them for a written explanation of their reasoning and have yet to receive one. So that is why, sadly, I cannot report back to you the remarkable individual data that the Barcellos lab produced. I could only show you the aggregate data, and I'm really, really sorry for that. So this brings us near the end, and let's return to the issue of a personalized medicine and what are the obstacles in the way. Well, one of the obstacles is the unresolved issue of who should control access to your genome. The CLIA amendment would have it that only your physician can give you access to your genetic information. Well, much as I love my personal doctor, there are MDs that have gotten their MD in this state, from reputable places like Stanford, that have not gotten a bit of genetics in their MD training. I don't think they're enthusiastic about interpreting my genome for me. I spent 20 hours on it. When I visit my physician, I get 15 minutes. Not much she can do. Now, there is a fantastic profession in this country of genetic counselors. There's a couple dozen programs in North America. They each train a handful of people per year. Really not enough to address the oncoming onslaught of very important genetic information that we have to have access to and be able to interpret on our own. This is a really important issue that needs attention. Of course, education is critical. The gap between what we thought people understood and what they really understood, boy, I'll tell you, I was really surprised by that. Personalized medicine implies making medicine specific for a subset of the population with different classes of a particular disease. Clinical trials are really expensive. It is not at all clear what the complications will be for the cost of clinical trials when the number of consumers for each of these drugs is smaller. Will the drugs become more expensive? Will it become more expensive to do the trials? Will trials not get done? These are important issues that will be worked out in the coming years. The FDA approval process probably needs attention, and we are at, I think, reasonable risk that the FDA will decide to regulate direct-to-consumer testing. There's certainly good things about that, but if their regulation requires that there be validated clinical guidance about the interpretation of any of the possible 9 billion variants in a genome, let me tell you, it's never going to happen. And that regulation would prevent you from having access for you to use your wit, your judgment in interpreting your particular genetics. And of course, there's the very real issue of genetic privacy concerns. Now, a small but important step has been taken with respect to genetic privacy, and that was, in my opinion, the best thing to come from the Bush administration. In 2008, the Genetic Non-Discrimination Act was passed and signed here. The vote in the Senate was unanimous. Somehow the House managed to drag up 3 people to vote against this. I don't know how. And this act, really importantly, prohibits genetic discrimination in health coverage or employment based upon genetic tests. It's good. It doesn't protect you from a requirement for genetic tests for long-term health care, and the Department of Public Health estimates that 1 of 6 of us will need that, and it doesn't protect against genetic discrimination in life insurance. So we have, as a culture, some ways to go. So let's take a look into the future and try to imagine what might happen in the coming years. And the future is always hard to guess, and it's really important to get the details right, as shown here on the slide. Now, what are my dreams of what's going to happen with genetic information? And I have to say that I have many dreams. The first of them sort of resonates with all of us, I think. I have a dream, and it's not too far off, that our individual nutrition can be optimized by knowledge of our particular genetic variants. I'm really looking forward to participating towards that goal. And as Francis Collins, the director of the NIH, has said, we don't have a healthcare system in this country. We have a sick care system. And I think knowledge of our personal genetic variants can help us recognize our risk factors before they are sick factors so that we can promote health. And promoting health is so much more cheaper than treating disease. This may be the single most important thing that we could hope for controlling the escalating costs of medical care in this country. But I have a bigger dream. My bigger dream is that some of the freshmen, as a result of this program, will be inspired to do something in the realm of personalized medicine, either as an MD or dealing with the social aspects of it, or actually coming to the laboratory and doing something amazing. Let me show you the footsteps that some of your predecessors as undergraduates here at Berkeley have done. Let me introduce you to the human gene called CCR5. There's the sequence of that gene written in the language of amino acid sequence. This gene is remarkable. We all have this gene, but in 11% of people from certain parts of Europe, there is a mutation in this gene that blocks the ability of HIV to infect cells. That's pretty amazing. It's not a perfect block, it's a pretty good block. So what can those of us that don't have that variant ever even imagine doing with that kind of genetic variation? Well, this gives me the opportunity to introduce Fyodor Urnov. Fyodor Urnov is a full-time senior scientist at a local company called Sangamo, and to our great benefit is also an associate adjunct professor in the Department of Molecular and Cell Biology and one of our favorite professors. Fyodor works at Sangamo, whose expertise is the development of these things, zinc finger nucleases, which are nanomachines for genome editing. It is possible to make a zinc finger nuclease these days that allow you to edit the sequence information essentially at any gene, probably in any genome on the planet. So if you think about this technology and the ability of mutations in CCR5 to provide HIV resistance, can we start to think about the C word? Can we start to think about cures? Well, here's a paper that gives me hope. It's a paper that shows with zinc finger nucleases, we can edit that gene and make HIV-resistant immune cells. Note the authors that are highlighted in yellow. All but one of them was a Berkeley undergraduate doing an internship at this company making this kind of a discovery. One of the authors is a Berkeley graduate student. Now, if that wasn't good enough, most recently we've seen that the same advance has been made in stem cells, so we can actually think about an age when somebody infected with HIV can have their own stem cells removed, genome edited, put back into their bone marrow, and reconstitute an HIV-resistant immune system. How would you like to have said you carved your initials on that kind of an advance? That would be stunning. Now, if you're interested in learning more, let me direct you to two books that are utterly fantastic. One of these is just recently out. It's by Francis Collins, the director of the NIH, called The Language of Life. It's all about the kinds of things we talked about tonight. This book is so good that I bought 100 copies of it to give out tonight, and any of you that have a number on your ticket ending in 6 should feel free to pick up a free copy. Once you've read it, give it to somebody else who might want it. This other book that's absolutely terrific, written by two colleagues, Stan Fields at the University of Washington and Mark Johnston, will be out shortly and would make a great Christmas present for any members of your family. Let me remind you that this is just the kickoff event in this year's On the Same Page program. Mark earlier described some of the events, but let me highlight a special interest I have in just a couple of these. One of the events of great importance is this direct-to-consumer DNA testing panel that will be in this building on Thursday afternoon. One of the participants in that is Professor Hank Greeley, who is one of the most outspoken opponents of what we did here at Berkeley. He's also the one opponent that I have the most respect for. Now, when he was invited to come here, he said, well, I'm happy to come, but he told our dean, he goes, you know, I don't expect any students to show up at that. And our dean assured him that that expectation came from the fact that he teaches at Stanford and that Berkeley students were really different. So I want this room packed with bears that afternoon. So don't let me down. I'm going to be there taking attendance. And finally, let me remind you of the presentation that will come from the incomparable Altacharo. And if you have never heard her before, you are in for a treat in this room, September 29th. Be there or be square. This program has benefited from so many people that I don't have time to thank individually, but let me just deal with the subset that it would be incredibly impossible to not highlight. And that is, first and foremost, the Barcelos Lab, who dropped all of their research, dropped everything else they were doing to help us get these gene tests done. So I can't thank them enough, and you saw how good of a job that they did. We had a lot of help from a lot of friends, but these three really stand out. My dean, Mark Schlissel, was there through thick and thin with us, and boy, I'll tell you, his support was absolutely indispensable for the success of this program, as was the extraordinary heavy lifting that Alex Schwartz and Nora Paymer did. Now, it's also true that this controversy on this campus has done a lot and has been noted worldwide. And I've gotten a lot of emails that have expressed from colleagues at different campuses that have expressed extreme amazement that we were actually even able to try something like this. And although we didn't get to the end in terms of delivering individual genetic information back to students, we have opened that door. And having opened that door, others are going to drive through it. And because of the incredible support from the leadership of this campus, and I know in particular Chancellor Bergenow, who in the midst of all of this was able to stand above the fray and say, this is what Berkeley is all about. This is Berkeley at its best. And that support has been really indispensable and inspirational to me personally. I want to also thank the Howard Hughes Medical Institute, in particular Robert Tejan, its president, who's also a friend and colleague here at Berkeley, a Berkeley professor, for the financial support to pay for all of the gene testing. And it's often been said that ignorance is bliss, but in the era of personal genetics, ignorance is never illuminating. So let me close with fiat lux and my deep appreciation for all of you that are participating at any and all levels in this program. Thank you so much for coming tonight. Thanks very, very much. Now, I know many of you have to go home and do homework, for example, but I would love to take the time to answer any questions that any of you have. There are microphones up here if you'd like to use them, and please identify yourself. I want to try to get to know as many people here as I can. So the floor is open for any and all questions.
Yeah, hello, my name is Vijay Gupta. My question is that the personalized medicine that you mentioned, I seem to has a fundamental contradiction in itself, in the sense that the current modality is that you test something on 1,000 people, excluding me, then apply the observations of that to me, thinking that I'm similar to them. But if I'm going to have a unique treatment that's unique to me, that means it has to be tested on me. So I'm always going to be a guinea pig for all the treatments that I receive.
That's a really important point. We are all different. We are all genetically unique. Even identical twins are genetically unique because they have about, on average, 80 base pair differences. So yes, the characterization of clinical trials is accurate. It's always an extrapolation based upon one population to people that were not in that population. But the goal is to try to understand the gene variants that provide the particular benefit or risk to people so that we can figure out more intelligently who should get which particular treatments. Very, very good point. Thank you, Vijay. Yes, what's your name?
Yeah, hi. My name is Liz Kearney. I'm a
Hi, Liz.
Hi.
What a pleasure to meet you.
Yeah, my name is Liz, and I am a certified genetic counselor and the president of the National Society of Genetic Counselors, and I appreciate the invitation to participate in this program, and also appreciate the opportunity to open up a dialogue about genetic testing and the positive and negative consequences that can come from that. And I wanted to take the opportunity to share with the audience some of the ramifications of, clinical ramifications of genetic testing that aren't as obvious when you, for a program like this, need to pick and choose, what tests you're doing. And one of the things you alluded to, I believe a little tongue-in-cheek, was Dr. Collins' comments about quibbling over clinical interpretation. And that particular example was that in sending his sample to 3 different labs, he received a result that from one lab was a high risk for the disorder, another lab that was an average risk, and another that was a low risk. And if you put yourself in the situation of a patient wanting genetic information that they would use for their health and potentially only getting one of those results, making health decisions based on that, that obviously has significant implications. And the advantages of working with someone like a genetic counselor who can explain that possibility exists can help to educate that patient. The other example I wanted to give, because you shared your own results, was the alpha-1 antitrypsin deficiency, which is a recessive disorder, as you explained, and causes a lung condition with emphysema even in non-smokers. And a colleague of mine recently spent hours on the phone with a patient who did direct-to-consumer testing with no prior counseling or any information, was found to be a carrier, and really, even though she was a bright woman, she'd gone online to find information and had difficulty consolidating it all, and was absolutely panicked by this result and thought that she was in fact going to get emphysema herself. And my colleague, I think she did an excellent job of educating that patient and got her eventually to a point where she really understood her information. But it is one of the risks, and when you're dealing with a situation where somebody has a family history or has a real life concern and you're applying genetic information, the opportunity to have a partnership with a healthcare provider who can ask the right questions, provide in-depth information, is a very powerful one. So thanks very much.
Oh, thank you, Liz. And something that you need to know is that when we were planning on being able to return genetic information, Liz and her organization had lined up volunteer genetic counselors to be available for any students who had any desire to have any genetic counseling on their results, and I thought that was an extraordinarily magnanimous offer on your part. Thank you. Question over here, your name.
Hi, Dr. Rine. My name is Jessica Rakiash. I'm actually a 4th year here, and I'm a molecular and cell biology major, and my emphasis is genetics. So I'm really all about this stuff. I actually took Bio 1A with you too, but
how could I forget?
So anyway, early in the talk you did mention something about the gap between technology and understanding.
Yeah.
And as a Cal student and as a genetics major, this is kind of a duh for me. I have no problem with this program, and I don't understand why people do. But a lot of the lay people who are not as educated as I am about things genetics related, how can you see that gap closing, and will it be in time for the true advent of personalized medicine?
Yeah, that's really the fundamental issue about how many thought leaders do we need to produce in the community that can vocalize clear and cogent explanations of the science against the concerns that come from the lack of understanding. So what I want to do is to teach genetics as a required course to all entering freshmen in the College of Letters and Science in the Greek theater, so that we can get 5,000 educated people out there. And Mark, I think you owe me one, so can you pull that off? Yeah, we depend upon people just like you to come here to learn this stuff, to understand the significance of it, and to be the spokesman out there. Absolutely. Thank you.
Hi, my name is Vincent Guaci, and I just kind of wandered onto the campus a couple months ago. And I had a question about the snake oil salesman. You pointed out you had the high-quality companies. So what happens to the snake oil salesman? What penalties are there, and how do we stop these people from scaring people amazingly by telling them results of horrible genes that will panic them?
Yeah, well, snake oil salesmen tend not to have as a profession scaring people. They have a profession of selling people something, right? So they tend to, and I'm generalizing, painting with a broad brush here, they tend to sort of raise the, sort of the worried well concerns, and then we'll want to sell you something to deal with that. Frankly, I don't have a good solution for that. I do think that some regulation would be a good idea. But what I am really worried about is that policy is a blunt instrument. And if we have a regulation that inhibits the development of the technology that lets us have access to information that really is enabling, that when properly interpreted can really be beneficial, then we all get hurt by the misadventures of the few bad guys. And that's a difficult problem, but I really think we've got to be very careful to be very surgical in the attack against that.
Right, so I think that's where the geneticists can play a big role and the genetic counselors to be involved in that, as opposed to leaving it just to the legislatures.
Yeah, thank you. I completely agree with you. Other questions or comments? Yes, shout it out.
Hi, I'm Claire. I'm a freshman, and I have a comment. You were saying that one of your goals was to raise awareness and stuff and inspire people, and I'm inspired. But I was gonna say that like just this whole program, the whole on the same page thing, it might not have worked out as we all didn't get our results, but it got me and my friends talking. Like I had some friends that I didn't even know were going to Berkeley. And like I slept over at my friend's house once, and in the morning I'm like, oh, what do you want for breakfast? She's like, I can't eat. I'm like, why can't you eat? She's like, I have to do my DNA testing. Because with the saliva thing, you weren't allowed to eat for however many hours before. I was like, oh, are you going to UC Berkeley? She's like, yeah. So like, so it was like a bonding thing, and we all started talking about that, and like people's parents started talking about it. Like some parents were totally against it, like, oh, UC Berkeley wants to get our genes on file. And other parents thought it was cool, and like we all started talking about it. So good job, you raised the way.
Yeah, thank you so much for that. That was exactly what our goal for this program.
We definitely talked about dog saliva.
So was that right? Was there 2 dog samples in there?
I was actually at my friend's house. We were gonna do it, but we're like, nah.
Ah, thanks so much.
Before we break, on behalf of the College of Letters and Sciences, we'd like to present Jasper with a gift for his leadership role in what we think is a spectacular educational program. One of Jasper's avocations, in addition to spreading the gospel of science, is he's quite an accomplished and avid sailor. So we've gotten him a ship's clock for one of his sailboats. And the inscription on the clock is Steadfast Captain of On the Same Page. So thanks very much to Jasper.
Well, thanks so much, and thanks for joining us tonight, and enjoy the rest of the program. It's really superb. Don't miss any of it. So long.